Hello, good evening, everyone. Am I audible?
Yes, sir, you are audible.
So, my name is Ubaid Tariq, and I welcome you all in the NPTEL problem solving session of the course named Cell Culture Technologies. This is the week 8 session of this course, and this will be the last session. Uh, this will be the last problem solving session, and you must have also completed the week 8 lectures, and as week 8 lectures are already live. So we will directly go to the questions and solve the questions. So first question is, during depolarization, membrane potential shifts from. The answers are positive, negative to positive, positive to negative, remains unchanged, none of the above. What is the answer? You can write in the chat box. So some of you have answered it correctly. As you know that in the depolarization phase, the sodium ions, they move inside the cell, and the potential, membrane potential, shifts from negative to positive. Another question is similar to this. During repolarization, membrane potential shifts from negative to positive, positive to negative, remains unchanged, none of the above. Yes, the answer is B. The action potential, it changes, or membrane potential changes from positive to negative during the repolarization. So I will explain this, what might is related to the questions. This is the representation or graph of the action potential. Here in the x-axis, the time is shown in milliseconds, and in the y-axis, voltage is shown. So here this is the zero voltage, and below this is the negative action potential or negative voltage, and above there is a positive voltage. So resting membrane potential in our neurons or other cells, it is minus 70. Minus 70 millivolts is the resting membrane potential. So the cells are in the resting membrane potential, or neurons are in the resting membrane potential when they receive the stimulus. So after when the cells receive the stimulus, the depolarization occurs, and depolarization occurs by the sodium intake of sodium ions into the cell. When these sodium ions go into the cell, earlier the charge inside was minus 70. Now, because of the sodium ions, positive charge of the sodium ions, the charge will abruptly go up to plus 40 millivolts. As there is a very fast influx of the sodium in the cell, so depolarization phase is, it is very fast. So here the depolarization, it also depends on the stimulus. If the stimulus is less, like here shown some of the failed initiations, like here the stimulus was very less, and there is some threshold which is minus 55 millivolts. When the stimulus is less than this threshold, so below the threshold, the depolarization or fast influx of sodium channels will not occur. And when the stimulus is pretty good, so it crosses the minus 55 threshold, so the abrupt influx of sodium ions occurs, and this depolarization occurs. And the question was asked, in case of depolarization, action marine potential or action potential, it changes from minus negative to positive. So the negative to positive is the correct answer. And in case of repolarization, here the cell is depolarized, now the cell is repolarized, and in case of repolarization, these potassium ions, they move out of the cell, or efflux of potassium ions occurs, and which cause the repolarization, and after the repolarization, uh, some cells will go for hyperpolarization, and then they come to the resting state. Here in this hyperpolarization, the balance of ions occurs, and after that the resting membrane potential of minus 70 is achieved. So here there are some of the things that I will explain in the next slide. So there are different types of action potentials the base of the cell, or on which on the base of cell there are different types of action potentials. For example, earlier I was showing the action potential of central nervous system, or the muscles, or the conduction, actually this one, it is the cardiac conduction, action potential of cardiac conduction. When the cardiac muscles receive the stimulus, how this action potential changes, what are the different phases. And in case of muscle cells it is different, and in case of neurons it is little bit different, and in case of here these are the cardiomyocytes, but there are the SA node cells. SA node cells also have the different action potential. So here talking about this cardiac action potential, here also the resting membrane potential is negative. It is and after the stimulus, this membrane potential it increases from a negative to the positive value due to the influx of sodium channels, and also the influx of calcium. But it is mostly because of the fast influx of sodium channels. And this here this red line is called phase 4, or resting membrane potential, and after the depolarization it's called phase 0. So when this depolarization occurs, there is another phase in which the sodium channels gets closed, and now the depolarization stops, and this here when this depolarization stops, then the another phase starts, which is the phase 2, and it's also called a resting membrane potential, or sorry, it's called a resting phase or plateau phase. Here no change occurs. It occurs because of the balance between the influx and outflux of potassium ions. There is the influx of calcium ions and outflux of sodium ions, and also some chloride ions also. But mainly these two ions are involved, and this lead to the formation of this refractive this period, or plateau phase. And here this in case of cardiac cells, this prolonged depolarization is distinguish cardiac muscle cells from the skeletal muscles. Here in case of here this very long refractory phase, or refractory phase, or this linear phase, it It only occurs in case of cardiac muscles, but if we talk about the skeletal muscle cells, or they have the very less this prolonged depolarization phase or refractory phase. Here in this phase or refractory phase, the if the second action potential is given or second stimulus is given to the cardiac muscle, it does not receive, or it does not the contraction does not occur, or the further depolarization will not occur because of the refractory period. And it is of the great importance in case of cardiac muscles, because if during this contraction period, if the another stimulus is received, and if here in the refractory period, if another stimulus is received and the cardiac muscle it detects, or it detects these stimuli, then it will lead to the inappropriate cardiac muscle contraction, which leads to the cardiac arrhythmias and a lot of cardiovascular disease. So this is very important, this refractory period. And after that, rapid repolarization phase, is it is same like the other cell types. Here also the potassium channel, if potassium efflux occurs through these channels, and it leads to the decrease in the voltage, and it is voltage is after repolarization, the resting membrane potential is achieved. And this is the whole action potential in which the cardiac muscle cell contract or skeletal muscle cell contract in this cycle. So now there is another question which is also related to the same thing. During depolarization, which ions move inside the cell from outside? What is the answer? I already explained this. Yes, the answer is A. During depolarization, sodium ions, they move from outside of the cell to the inside of the cell, and which leads to the depolarization phase. This one I already explained in earlier slides. So question number 4 is, which units of muscle cannot be used for force measurements? So as the muscles contract, so muscles have the muscle cells present in the muscle tissue. When they contract, the contraction force is measured uh, using various techniques. So which of these cells which are mentioned below is a basic unit of the muscle, and which is the unit of the muscle but it cannot be used for the force measurement? So the answers are myotube, myofiber, myoblast, none of the above. Yes, answer is C. So myoblast, they are actually the progenitor cells. They do not have the contractile power. So myoblast, they are not used for contractile measurements. For example, my here contractile measurements with respect to the cell. Here, you can say that the question may arise in your mind that the the contraction of the tissue should be made, for example, whole muscle or uh, a whole cardiac thing. But here the cells contraction of cells is measured. So there are different types of cells, and among them myoblasts, which are the progenitor cells of the muscles. They do not uh, they are not used for the force of contraction measures because they do not contract. So here in this slide I have explained it. So how I have in this slide, this is the slide of myogenesis, how the muscle fibers or muscle cells are formed. So there in our body, the satellite cells are present, and these satellite cells, they are usually in the quiescent stage. These quiescent state cells, they are not active, but in case of myogenesis, or when there is a damage to the muscles, so these quiescent satellite cells, they are activated, and after activation, they active satellite cells are formed. And these active satellite cells, after activation or after self-renewal, they proliferate. And after proliferation, these myogenic progenitor cells or myoblasts, they are formed. As I already said in the question, these myoblasts, they are the progenitor cells and they do not contract. But as they are the stem cells, they are the progenitor cells. And these progenitor cells, they differentiate into myocytes, and after that these myocytes fuse and from the myotube, and now this myotube has the contractile power, and these myotubes then mature, and after maturation these myofibers are formed. So in myotubes and myofibers only there is a contractile power, and usually myotubes are used to study the contraction of the cells, or if we want to study different drugs also. So these cells are seated on the cantilever, and when we apply different types of drugs, or if we want to discover some drug which has a effect on muscle contraction, so these drugs can be given and the contraction can be studied. So moving to the next question. Next question is neuromuscular junction is made at the junction of. So where this neuromuscular junction is present. So it is present at the sum of answers are given, like A part is muscle and motor neuron, glia and motor neuron, hippocampal neurons and osteoblasts, and none of the above. Anyone knows the answer? Yes, Sandhya. Sandhya has answered it correctly. Am I audible? I think there is some network issue. Sorry, I lost my connection. Am I audible now?
Yes, sir.
Yes. So, I was here in this question. So neuromuscular junction is made up of is made uh, neuromuscular junction is made at the junction of. where it is present at the junction of muscle and neuron, glia and motor neuron, hippocampal neuron, osteoblast. So anyone have answered it? Yes, I think earlier Sandhya have answered that the answer is A, muscle and motor neuron. So these neuromuscular junctions or neuromuscular junction, it is a synaptic connection between the terminal end of the motor neuron and the muscle may be the skeletal muscle, smooth muscle, and cardiac muscle. These are all the three muscles which contract, and they receive the motor innervations from the brain for the contraction to control their contraction. And it is the and this here the here the this synapse, it's called the neuromuscular junction, and as it connects the nerve to the muscle. So it is the site of for the transmission of action potential from the nerve to the muscle, and it is also the site for many diseases and the site of action for many pharmacological drugs. Why it is the site for many diseases? In case of many diseases, this there is effect on this neuromuscular junction, and the synapse here is affected. So the transmission of the motor signal from the neurons to this muscle does not occur, which leads to some disease. And in there are some drugs also which act in this neuromuscular junction, and upon acting on this neuromuscular junction, they produce some effects which will be beneficial for the patient. And how this neuromuscular junction is different from other synaptic junctions, like you know that the every nerve cell it connects to the different nerve cell using the synapse, and that synapse is also similar to this neuromuscular junction. But here it is the nerve end, and now there is no further nerve cell here. There is the muscle is directly present, or the effector tissue is directly present. So here it is different. It is different here, like it is. This is the presynaptic. In case of normal synapse, this is the presynaptic signal, or presynaptic neuron. Here is the synapse, and here is the postsynaptic neuron. But here are different components. Like this is the nerve terminal, and as this is the nerve, this is not called the presynaptic, as it is the terminal end of the neuron, so it's called nerve terminal. This space is called synaptic cleft or junctional cleft, and here the muscle which has the receptors for the these neurotransmitters which are secreted from here, and this is called motor end plate, the effector organ, effector muscle. So nerve action when the motor neurons give the nerve action potential, and these neurotransmitters, like the example here is acetylcholine, it's released and it acts on the receptors, and after that the action potential is generated in the this muscle. It may be the skeletal muscle, heart muscle, and cardiac muscle. After that, after the action potential is generated, which leads to the contraction of this muscle. So our question was related to this neuromuscular junction only, why it is present. So it is present here between the muscle and the motor neuron. So another question is, bungarotoxin causes paralysis by blocking potassium channel, acetylcholine receptor, both A and B, none of the above. What is the answer, anyone? So the answer is acetylcholine receptor. Here I will show it again. Like earlier, I was showing you. This is the nerve terminal. This is the synaptic cleft or junctional cleft, and this sorry, what was it? It was nerve terminal, and it was motor end plate. This was a motor end plate, and there are some receptors like receptors for nicotine, acetylcholine. There are nicotinic receptors. There are muscarinic receptors which are present here in the postsynaptic or this no, this end. So this what is this alpha bungarotoxin or bungarotoxin is it binds to the this receptor where actually the acetylcholine binds, and it prevents the transmission of signal from the neuron to the muscle, and which leads to the muscle paralysis, and that's written here alpha bungarotoxin. It binds to the actually this nicotinic acetylcholine receptor, and it prevents acetylcholine from binding to this receptor. And this receptor, as you know, that it is this receptor is present in the postsynaptic side of the neuromuscular junction, and and the function of this actually neurotransmitter was to attach to this receptor, and the action potential it is now it is generated in the muscle. But because this alpha bungarotoxin binds here to the receptor, the acetylcholine will not bind, and the signal transmission does not occur. So, because of the binding of bungarotoxin, the inability of acetylcholine to bind to its receptor prevents sodium channel from opening in the receptor. As you know, that sodium channel opening is the first step of the action potential. The action potential from the presynaptic neuron is blocked, and the muscle contraction is blocked. So, action potential does not transmit to the this muscle, and this leads to the paralysis. So there are other toxins also. Some may bind to the postsynaptic, some may bind to the presynaptic neurons, and they will lead to some defects or some diseases. So another question is for myotubules cultured on cantilever devices. To move cantilevers, which property of cantilever should be similar to myotubules? As I already said, myotubules they are cultured in the cantilever to check the contractile properties of the myotubules, or to check the if to check the contractile ability or contractile function of different drugs. For example, some group have discovered the drug which they propose or hypothesize that it has a function like it can enhance the muscle contraction or heart contraction or smooth muscle contraction. So if that drug needs to be checked before the before animal model, if it needs to be checked in vitro, so this cantilever is this cantilever device, the cells are cultured, and usually in case of muscles, these myotubes they are cultured to check the contractile power of these cells. So, what properties should this cantilever be should have so that it can be used? Actually, the properties of the material, mm, the material from which this cantilever should be made, what properties it should have similar to, what properties it should have similar to myotubes. And the answer is four answers are given. A part is stiffness, B is color, C is weight, and D is width. Anyone? Yes. So the answer is stiffness. Why so? Because the color and weight it doesn't matter, but in case of to check the contraction, stiffness of the material on which the cells are seated, it matters. if this cantilever device on which the cells are seated, it has more stiffness as compared to myotubes, so it will hamper the contraction or it will give the wrong result. So it should be similar. This cantilever should have similar stiffness and it should be related to these myotubes. It should have similar stiffness to myotubes. And usually these cantilever, they are made up of different soft materials like silicon. Usually they are made of silicon. And here in this slide I will show you how this cantilever works. Here this is the cantilever. Here it is placed on this platform, and this cantilever, it is the myotubes are seated in this cantilever, and uh, because of the contraction of these myotubes, there will be the deflection. If this contract, it will deflect. I will clearly show in the next slide. And the laser light is irradiated on this cantilever when this deflect, so the path of this laser light, it will change. So, so the contraction can be measured how much this cantilever is contracting, and it's contracting because of the myotubes which are present in this. And if some drug treatment needs to be given, so here the drugs are injected here in this, so and in institution, and the effect of that drug on the contraction should also be detected. So here I have explained the same thing, like these myotubes, if when they are seeded or they are cultured on this cantilever, and after that the contraction studies are done. For example, in case of when these cells contract, this cantilever deflects, and because of the deflection of this cantilever, uh, uh, and actually the laser light is falling on this, and the detector is detecting the laser light, and due to the deflection, the contraction can be measured using the photo detector. How much actually it is deflecting, it can be measured. So another question is, hippocampus is associated with movement, color, learning and memory, vocation. So what is the answer? Actually, I think this question has been repeated several times, maybe 3 times in 3 different sessions. So the answers are movement, color, learning and memory, vocation. What, anyone have given the answer? Yes. So the answer is learning and memory. So I have earlier also shown you similar type of slides. So this is the place of this hippocampus where it is present. So here this is the hippocampus, and here it is highlighted where it's actually present inside the brain and inside the lobes. And the main function of hippocampus is it senses the smell, uh, memory formation, intellectual thinking, sexual arousal, behavior, emotions. These all are regulated by the hippocampus, and the major function among them is the memory formation, long-term memory formation, and the formation of location-based memories or imagination. All these memories, location-based, imagination-based memories, these all are sensed by this hippocampus. So another question is, dash is the pungent pain producing compound found in the plants of the capsicum family. Exerts it exerts excitatory, desensitizing and toxic effects on a subset of sensory neurons, including the polymodal nociceptor population. So, which one of this is the answer? A part is the capsaicin, B part is TTX, and D is digitoxin, and D part is xanthine. Anyone have answered? Yes, the answer is capsaicin. Actually, this capsaicin, it is a compound of the chili. So here is the chemistry of chili. So there are some of the common capsaicinoids which are present in the chili. The most common among them is the capsaicin, which is uh, which is responsible for the bitterness that you feel when you eat the chili. And there are another component, dihydroxycapsinin. So the spiciness of the chilies is due to the presence of these capsaicinoids, capsaicin and dihydroxycapsinin, and these two compounds are main capsaicinoids in the chili pepper, and they cause burning sensation when they come in contact with mucous membrane due to their interaction with the pain or heat sensing neurons. So capsaicin is also used uh, in the pepper spray and other things, and it is also explored for its physiological effects, like in case of cancer or other things. And here in the right side there are different types of capsaicins and the index of how much beta they are or how much irritation they cause, it is mentioned. And these capsaicins actually they cause irritation on the mucous membrane, or if they are sometimes rubbed on the skin, and they also may cause irritation. So another question, or last question, is dash dash blocks sodium channels: TTX, 4AP, calcium, free radicals. So which one of them is the sodium channel blocker? Yes, tetrodotoxin, TTX. So there are a lot of calcium channel blockers which block the calcium, which block the sodium channel, and these they block the sodium channel and they interrupt the action potential. And some of these sodium channel blocks they are also used in case of anesthesia, local anesthesia or central anesthetics. They are used. And what actually these TTX they will block the voltage gated sodium channels, and due to this voltage blocking of these voltage gated sodium channels, so the action potential will not be generated or it will not be transmitted from one neuron to the another neuron. So this will be hampered. So it this TTX, it has been also used. It may be used to slow the pain transmission. Actually, when there's a pain, the signal goes from these in the from the motor neurons and to this I think through the sensory neurons, sorry, and it will be detected and you feel the pain. But because of the sodium channel blockers like TTX, they will block and this pain transmission can be stopped, so that the person does not feel the pain. So this was all about the today's session, and also this cell culture technologies. You have also completed the course. So this will be the last session of this course, last live session. You may have some maybe assignment date is still not completed. You have some days for the assignment, and after that you can appear for the exams. So this is my YouTube channel. Maybe after completing these sessions, or maybe sometimes I will upload some of the lectures here. So you can subscribe to these channels to remain up to date if I upload some of the lectures there. And also these lectures I record them, and I have already uploaded all of them in this channel. So you can search this channel in your YouTube. And thank you all of you for joining my all of the sessions. Some of you were quite regular and they joined all the session. Thank you so much to them and all others which have joined today's session. So if you have any questions, you can ask related to today's session, or any other questions related to your course. So if there are no questions, I can conclude this session.
Sir, excuse me, sir. Is it
Yes.
possible to get model question paper, sir,
Model paper.
for the exam, sir?
I don't have any idea about the model paper. But what you can do, you can ask in the discussion form of the course.
Okay, sir.
So
Thank you.
actually that your this exam, it will be related to your courses. So you study your courses well, you will easily qualify. So
Okay,
sir,
thank you, sir.
okay. So Sandhya has asked, dash cells are derived from the isolated inner cell mass (ICM) of the blastocyst stage embryos. So I don't exactly remember the answer now, but maybe but we don't have next session. So what I can do, I will answer you in the YouTube channel. When I upload this video in the comments, I will answer you. I will give the answer to this question. So if you further have any questions, maybe related to the course later, so you can ask me in the YouTube channel. You can write in the comment. I will surely answer you. So through the comment section, you can contact me actually. So thank you so much. Thank you everyone for joining all of these sessions. It was very nice to interact with you, and it was very nice to give these sessions. And thank you so much. All the best if you are taking the exams of this course. Thank you so much.